Welcome to our lecture on cell transport, diffusion, osmosis. I'm really excited to talk about this because it's a super important subject and it's keeping you alive right now, so it's a big deal. I'm going to pause the video and point my camera downward so I can start drawing things out for you. Ready? Alright, so we're going to be talking about transport. Super exciting. It's a really big subject. And when we talk about transport, there are two types of transport. There's active transport and then there's passive. Active transport is one type, passive transport's the other type. I'm going to call this a flow chart, by the way, because it's not really a concept map since I'm not attaching a description for how these two things are related over the arrows. Instead, I'm just adding arrows. So more of a flow chart than a concept map. And we're just going to try and organize our thoughts about the differences between active and passive transport, knowing that those are the two types of transport there. So the main difference is that active transport, it requires ATP. I know that you know what ATP is a little bit because you read about it in the cell chapter. ATP is cellular energy. It's what your cell uses to do things. Now when we think about active transport, there's a couple different types of active transport. One is electrochemical gradients. Electrochemical gradients. This is really important because this is how your muscles work. And remember, your heart's a muscle, so all of your vital organs are using muscles as well. So this is keeping you alive right now, active transport. And this is also how your neurons work. So you thinking about these concepts right now is using active transport in an electrochemical gradient. Another type of active transport that's important for this class, endocytosis is going to be taking things into the cell. Then there's two types of endocytosis. If you're taking a solid in, then you call it phagocytosis. If you're taking a liquid in, then it's called pinocytosis. Which I remember pinocytosis is liquid because Pinot is a type of wine and you drink it. Solids is phagocytosis. This refers to engulfing something, so you're engulfing something into your cell. But both of those are your two types of taking things into the cell. If we're going to use energy to take something into the cell, we're also going to use energy to take something out of the cell or to push it out of the cell. That's exocytosis. Cytosis refers to cell. Exo means outside, like exoskeleton. So outside of the cell, ejecting something from the cell. Now, if you read your book really closely, you probably noticed that there's another type of endocytosis that I didn't mention. It's not just pinocytosis and phagocytosis. It's also cell-mediated, receptor-mediated endocytosis. But that one we're not going to talk as much about, and it's less important in your daily life, so we're just going to focus on these main two. So we know that active transport requires ATP, and that there's three main ones that we care about. Something else that's important for active transport is active transport, adding another one down here, can move things against its concentration gradient. Now that against is different. That's not really a normal thing for a cell to do, because cells like to move things in order from high concentration to low concentration. This is going to move something from low to high. Low concentration. I'm going to abbreviate that to high concentration. So it's going against its concentration because it's going from where there's a low to where there's a high. It's making it more crowded in a sense. I'm going to pause it and get another piece of paper and keep going. Okay, so I folded my piece of paper in half so I have more space to continue. And we're going to keep talking about transport. So we know active transport requires ATP and can move things from low concentration to high concentration. But passive transport's really what we want to talk about today. That's because passive transport has two main types, and it is going to be diffusion, and the other main type is going to be osmosis. We know that active transport uses ATP, so what makes passive transport passive is it does not require ATP. This is basically free transport. It doesn't have to use anything for this to happen. So if you're not using energy to make this happen, you might be curious why it happens at all. And the answer to that is molecules move. So all the molecules in things like in liquids or gases or solids, they're bouncing around and jiggling, and eventually they'll slowly knock into each other, kind of like billiard balls will knock into each other, and they'll push each other around. So it's the subatomic movement of molecules that causes diffusion and osmosis to occur. You don't have to pay for it with ATP. It's just going to happen because these molecules have energy and they're jiggling. So they're going to jiggle themselves into different places than when they started. Let's talk first about diffusion. Diffusion is the movement of a solute from a high concentration to a low concentration. So the movement of a solute from high concentration, I'm again going to abbreviate that, to low concentration. This means you're going from where there is more of a solute, where the concentration of a solute is higher, to where there's less solutes. We also say this is with the concentration gradient. Remember that when we were talking about active transport, we said it goes against the concentration gradient. In diffusion, you're going with the concentration gradient. So that's what diffusion is. Now, if you have diffusion just in the air or in a liquid, then we have simple diffusion. Like when you drop a tea bag into a hot cup of water and the tea brews, all of the tea that was concentrated in the tea bag just slowly moves out equally in the water. That's simple diffusion. If you have diffusion that occurs across a membrane, that's going to be facilitated diffusion. So this occurs across a membrane. Now, if it occurs across a membrane, most molecules can't go just through the plasma membrane. Small things like oxygen gas and carbon dioxide gas and tiny little non-polar molecules, they can. But anything that's got a charge, any ions, or anything that's polar, or anything that's kind of bigger like a sugar, all of those things are way too big to go across the membrane on its own, and so it's going to use a protein. The protein that it's going to use are called transport proteins. Transport protein. Scooch this up. We have two main types of transport proteins. We've got channels, and then we've got carriers. Channels, they kind of look just like a tube going right through your membrane. So whatever you're going to try and move can just go straight through. That's a channel. Carriers, they're going to move and transport your solute. So sometimes they look like, like a puzzle that's missing its neighbor. Here's my membrane on either side. And then your solute will pop into this little puzzle hole, and your carrier will slowly push it through. So that's step one, it pops into the hole. Bloop, there it is. Next step, the little protein is gonna like suck it through. So here it is. And then the last step, your little carrier is going to pop it out the other end. The main difference between a carrier and a channel is that a channel is open all the way through. It's just open space, where a carrier is going to change shape to move your solute through. Cool. So that's facilitated diffusion. Happens over a membrane. Simple diffusion in liquid or air, but no membrane involved. Fabulous. Now that we've got diffusion, one type of passive transport done, let's look at osmosis. All right, osmosis. Osmosis is a type of diffusion, but it's the diffusion of water. I'm going to abbreviate water. Just water. Water is the only thing that moves, and it is always going to go across a membrane. Sometimes the membrane is dialysis tubing, so it's man-made. Sometimes it's a cell membrane, or a plant, or a fungus. But whatever it is, it's the movement of specifically water across the membrane. So that is osmosis for us. Now, when we're talking about osmosis, we have some fun vocab to add. I'm going to scooch this up a little bit. Here we are. I've got a beaker, yeah. Here's my fabulous beaker. My beaker's full of water. And inside my beaker, I have a cell. Y'all with me so far? So here's my beaker, there's my cell, my beaker's full of water. If I know that the solution outside has 10% sodium chloride, and I know that the solution inside the cell has 20% sodium chloride, I now have all the information I need to think about osmosis and figure out what's going to happen with the movement of water. So wherever you have a greater concentration of solute, remember solute is the thing that's dissolved. You say that it is going to be more solute concentration, and this is less solute concentration. So what's not written here is what the other 80% of the inside of this cell is. I can assume, because it's a biological system and this is a biology class, that the other 80%, so we get up to 100%, is water. In this case, water is the solvent. The solvent is whatever dissolved the solute. So if I make a cup of iced tea, the tea is the solute. The water is the solvent. However, if I'm going to marinate a piece of eggplant in olive oil and rosemary, the rosemary would be the solute and the olive oil would be the solvent. So solute's what's being dissolved, solvent's what's doing the dissolving. Here I know I have 10% of my solute, in this case sodium chloride. That means that the other percent to get up to 100, so 90%, is going to be water, which is my solvent. Now we know that in diffusion, you go from high concentration to low concentration. And osmosis is the diffusion of water. So I'm going to have my water move from wherever there's a higher concentration to a lower concentration. So the water is going to move from the solution. This pin is blue to represent water. Going from higher concentration, 90% outside, to where there's 80% inside, until the two are equal. So my net water movement is going to be inside the cell. Now, most osmosis problems are not going to give you the water concentration. It's just going to give you the solute concentration. So when you're looking at this problem again, it might look to you like in my beaker, I've got a cell in water, and water moves to wherever there's the higher solute concentration, which is true, but it's not that the water is necessarily attracted to the solutes. Instead, the water is moving along its concentration gradient. So wherever there's more solute, it means there's less water. So the water is going to flow from high to low, which to us looks like it's going toward whoever has more solute. Now what's cool about this too is we can add a couple extra vocab words. When we're comparing two solutions, the solution inside the cell and the solution in the beaker, we get to use our hyper, hypo, or isotenacity words. So if there's more solute, like inside this cell, we say it is hyper. Hyper means more. Tonic. There's more solute, that's what tonic means, inside the cell. And in comparison, because there's more inside, there must be less outside, which is hypotonic. Hypo means below. A hypodermic needle goes under your skin. A hyperactive person has a bunch of energy. So that's where those two words come from. And they're only used in comparison of each other. So if I didn't have a cell and I just had a solution that was 10% salt, I couldn't use those words because I'm not comparing two things. You can't say it's more than something if there isn't another something to compare it to. And if these two solutions had exactly the same amount of solute, then you use the vocab word isotonic. Iso meaning the same. They have the same tenacity. Oops. So, the way that these diffusion osmosis problems usually work is you're given a setup which is something like a cell is in a 75% sugar solution. The cell is 10% sugar. Which way will water move? Due to osmosis. Giving you a moment to think about it. I personally always like to draw it out. So when I would solve this question, I would draw myself a solution in a beaker. I know that the cell is in a 75% solution, so that means that the outside of the cell has to be 75%, and the cell is 10% sugar, so I've got 10% inside here. That means that 90% of the cell is water, and over here only 25% of the solution is water. So there's a lot more water inside than outside, meaning the water will leave the cell. My cell will start to shrivel up and shrink. We call that crenation. If the opposite were true, and I had my cell 75% sugar, and the solution it was sitting in was 10%, now most of the water is outside the cell. The water would flood into the cell, and your cell does not have a cell wall, so it might pop. If it pops, we call that lyse, which, by the way, is where the word Lysol came from. If you're going to buy that cleaner, you want to kill bacteria, you're going to try and lyse them. Lysing them means you're popping that cell.